Good morning, students. In previous class, we completed about the biomolecules. Introduction to biomolecule. What is biomolecules? Carbohydrate. Classification of carbohydrate on the basis of product on hydrolysis, monosaccharide, oligosaccharide, polysaccharide. On the basis of taste, sugar, non-sugar. On the basis of reducing nature, reducing sugar, non-reducing sugar after this one. So in second class of this chapter biomolecule, today we discuss about amino acid. Come to the point. Point one is, first one is amino acid. What is amino acid? Amino acids are the proteins. Normally proteins are linear polymer of alpha amino acid. These are regarded as building block of protein. Alpha amino, and that is amino acids are regarded as building block of proteins. These are represented as R, CH, NH2, COOH. It is alpha position, where these are represented as hydrogen or alkyl group or any aromatic ring. And this, where I repeat again, these are either maybe a hydrogen atom or maybe alkyl group or one may be aromatic ring. That means each amino acid is a nitrogenous compound containing both acidic carboxyl group and a basic amino group. It is basic nature amino group and it is acidic nature the carboxyl group. It is classified. This amino acid is classified into two types. One is neutral acidic and a basic amino acid. First one is classified as neutral acidic and a basic amino acid, and point two is essential and non-essential amino acid. Non-essential amino acid. We discuss one by one. First one is neutral amino acid, acidic amino acid, and a basic amino acid. Depending upon the number of amino group and hydroxyl group. I repeat again, the formula is R, CH, NH2, COOH. Depending upon the number of carboxyl group and amino group, it is divided into neutral amino acid, acidic amino acid, and basic amino acid. Point one is neutral amino acid. If the amino acid contain one amino group and one carboxyl group, then it is known as neutral amino acid. On the other hand, if the amino acid product contain one amino acid, one amino group and two carboxyl group, then it is known as acidic amino acid. On the other hand, if the amino acid contain one carboxyl group and two amino group, then it is known as basic amino acid. So point two, next is essential amino acid and a non-essential amino acid. Mark that those amino acids are not synthesized by the human body, and those amino acids are not synthesized by the human body, and it is necessary in the form of diet, then it is known as essential amino acid. For example, valine, leucine, isoleucine, valine, leucine, isoleucine, etc. And second one is non-essential amino acid. Those amino acids which are synthesized by the human body, not necessary in the form of diet, these are known as non-essential amino acid. For example, glycine, alanine, and tyrosine. For example, glycine, alanine, and tyrosine. These are some examples of non-essential amino acid, and these are some examples of essential amino acid. Next we discuss about peptide and polypeptide. Next is peptide and polypeptide. Second one is peptides and polypeptide. Mark that. What is it? These are the condensation product of two or more amino acid. The bond between two nearest amino acid is a special type of amide bond known as a peptide bond. The special type of amide bond is known as peptide bond. Special type of bond is known as peptide bond, and the chain thus formed is known as a peptide chain. The chain thus formed known as peptide chain. Peptide chain. Hence, depending upon the number of amino acid taking part in the condensation, peptides are classified as dipeptide, tripeptide, and polypeptide. Classified as dipeptide, tripeptide, and polypeptide. I repeat again. Depending upon the number of amino acid taking part in a condensation, peptides are classified as dipeptide, tripeptide, and polypeptide. First one we discuss dipeptide. That means if there are two amino acids joined together resulting in the formation of a product is known as a dipeptide. If three amino acids joined together resulting in the formation of a product, that is known as tripeptide. If four or more than amino acids joined together, then it is known as polypeptide. Proteins are generally polypeptide containing at least 100 or more amino acid. I repeat again. Proteins are the polypeptide containing at least 100 or more amino acid. Next, third one we discuss about the protein. Third one. Third one is protein. What is protein? Protein, these are the linear unbranched polymers of alpha amino acid. These are, I repeat again, these are the linear unbranched polymers of alpha amino acid. In a protein molecule, alpha amino acid joined together by a peptide bond formed between amino group and carboxyl group of a successive amino acid. I repeat again, in a protein molecule, alpha amino acid joined together by a peptide bond formed between amino group and carboxyl group of a successive amino acid. So these are the proteins are the essential component of a living cell and are essential to cell structure and cell function. Hence, we conclude that proteins are vital chemical substance and essential for growth and maintenance of life. I repeat again, for that reason, protein are the vital chemical substances essential for growth and maintenance of life. Next, we discuss classification of a protein. Next is classification of protein. Classification. Proteins are classified on the basis of two categories there. On the basis of molecular structure, protein is classified as fibrous protein and globular protein. On the basis of molecular structure, proteins are classified as fibrous protein, and point 2 is globular protein. We discuss one by one. First one is fibrous protein. What is a fibrous protein? These are the linear thread-like molecule having very strong intermolecular forces of attraction. I repeat again, fibrous protein are the linear thread-like molecule having very strong intermolecular forces of attraction, and they are insoluble in water. That is, the fibrous proteins are insoluble in water. For example, keratin, collagens. Example of fibrous protein is keratin. For example, keratin, collagens, myosin, etc. Next point to the globular protein. What is a globular protein? These are highly branched, cross-linked and are soluble in water, acid as well as alkali. For example, enzyme, albumin in egg, hemoglobin. Example of globular is albumin, hemoglobin, enzyme, albumin in egg, hemoglobin, etc. Fibrous protein is insoluble in water, globular protein is soluble in water. These are next. Second category, it is the first category, is their classification on the basis of molecular structure. Then we go to the second category, that is on the basis of chemical composition. On the basis of chemical composition, on the product, they give upon the hydrolysis. There are two structures there. One is simple structure, one is conjugated structure. That means to discuss on the basis of product on hydrolysis, they give two structures. One is a simple structure and one is conjugated structure. We discuss one by one. First one is simple structure. What is a simple structure? That means they give the amino acid. The simple structure gives the amino acid on hydrolysis. Examples are albumin, keratin, globulin. Example is, for example, albumin, keratin. Example of simple structure is albumin, keratin, globulin, etc. Next, we have to second one is, that is conjugated structure. These are the simple protein bonded with non-proteinous prosthetic group. Bonded with non-proteinous prosthetic group. Non-proteinous prosthetic group. For example is hemoglobin, chlorophyll, serum protein. Example is, for example, hemoglobin, serum protein, etc. Next, next we discuss about structure of protein. Next we discuss structure of protein. Next is structure of protein. Generally 4 structures are there. Then we discuss about structure of protein. The actual structure of a protein or the complete structure of a protein is quite complex. Generally, it is classified into 4 different levels. That is, one is primary structure, one is secondary structure, one is tertiary structure, one is quaternary structure. We discuss one by one. First one is primary structure. It refers to the sequence in which various amino acids joined by the peptide linkage. I repeat again. Primary structure refers to the sequence in which various amino acids are joined by peptide linkage and it form the backbone of the protein. The primary structure indicate it form the backbone of the protein. Next we go through the secondary structure. It refers to the arrangement of a polypeptide chain. It refers to the arrangement of polypeptide chain into three-dimensional structure of a protein assume. So depending upon the alkyl group, depending upon the side chain of the alkyl group, the secondary structure it may be of two types. One is alpha helix structure, and one is beta plated seed-like structure. Beta plated seed-like structure. If the size of the alkyl group is large, then it is known as alpha helix structure. If the size of the alkyl group is small, then it is known as beta plated structure. Next we go through the tertiary structure. That is primary over, secondary over, tertiary structure. It refers to the final three-dimensional shape. It refers to the final three-dimensional shape that result from the twisting, bending and folding of the protein helix within the forces operating hydrogen bond, disulfide bond and van der Waals bond. The forces are operating here, that is van der Waals bond, disulfide linkage, hydrogen bond, etc. This type of bond observed in case of the structure. I repeat again. The tertiary structure refers to the final three-dimensional shape that result from the twisting, bending and folding of the protein helix with forces operating hydrogen bond, disulfide linkage and van der Waals forces of attraction. Next we will go through the quaternary structure. Quaternary structure refers to the way in which the polypeptide chains of a complex protein are associated with each other. I repeat again. Quaternary structure refers the way in which the polypeptide chains polypeptide chain of a complex protein is associated with each other. Next we discuss the the important term for council exam point of view, that is denaturation of protein. What do you mean by denaturation of a protein? Next, very important is denaturation of a protein. What do you mean by denaturation of a protein? The process after which protein lose their physiological activity and certain other processes. I repeat again. The process after which protein lose their physiological activity and some other processes and is caused by bacteria, action of heat and also reacting with alkali, acid, alcohol is known as denaturation of protein. I repeat again. The process after which protein lose their physiological activity and some other process and is caused by bacteria, action of heat and also alcohol with acid, these are known as denaturation of protein. Example is, that means formation of cheese. Example is, first example is formation of cheese, heating of egg. It is known as denaturation of protein. Next we discuss about next point is enzyme. Next we discuss enzyme. What is enzyme? Next we discuss enzymes. What is enzyme? Enzymes are the complex nitrogenous organic compound. Enzymes are the complex nitrogenous organic compound produced by living plants and animals. These are simple biocatalysts. Very important one. Enzymes are simple biocatalysis or biocatalyst. Then we discuss about their characteristics of enzyme. Very, very important one is their characteristics of enzyme. 3 to 4 points are there. We discuss one by one. Point 1 is enzymes are highly specific in their action on substrate. Particular enzyme is fixed for a particular substrate. Point 2 is small quantity of enzyme needed to catalyze a large quantity of substrate. I repeat again. A very small quantity of enzymes needed to catalyze a large quantity of substrate. Next is they are optimum temperature. They have an optimum temperature at which they work best. I repeat again. They have an optimum temperature at which they work best. Next point is that is they work efficiently at a constant pH. They work efficiently at a constant pH. Example, near about the pH is 5 to 7. Then we discuss about some examples of enzyme in day-to-day life, how it acts as a catalyst. Example is, point 1, 2 to 3 examples are there. Point 1 is inversion of cane sugar. Discuss. That is, first example is inversion of cane sugar. That means we know cane sugar on hydrolysis give rise to glucose and fructose, and it is in case of enzyme is invertage. That is C12H22O11 plus H2O. Invertage give rise to glucose plus fructose. It is glucose, it is fructose, it is sugar or sucrose. Here enzyme is invertage. Second example is that is conversion of glucose to ethyl alcohol. Here enzyme is zymase. Conversion, second example is conversion of glucose to ethyl alcohol, that is C6H12O6. That is conversion of glucose. The second example is conversion of glucose to ethyl alcohol. Here the enzyme is zymase. Zymase, it convert to 2-ethyl alcohol plus carbon dioxide. Another example, next example, it is conversion of glucose to ethyl alcohol. Next example is conversion of starch to, so many other examples are there. For example, conversion of starch to maltase. Here the enzyme is diastase. Similarly, conversion of maltose to glucose. The enzymes are maltase. So many other examples are there. Last we discuss about application. Application of enzyme. Application of enzyme. 2 to 3 points are there. First application of enzyme. That is, first application is formation of beer, wine. Formation of beer, wine by the help of enzyme. Next is formation of medicine, life-saving medicine, that is insulin. Formation of life-saving medicine, insulin. Then penicillin. Then formation of cheese. These are some examples of enzyme. I repeat again, the formation of beer, wine, then formation of life-saving medicine, that is insulin, penicillin, formation of cheese. These are some examples of enzyme.